Hi, everyone. My name is Tony Zhang, and I'm the principal scientist and senior project manager of Creative Biostructure. It is my great pleasure today to introduce you the collaboration between Creative Biostructure and HiQ FATHOM EPR technology company. We have announced our collaboration in May. Now we are giving a webinar to our customers to highlight our collaborations and what essentially the services can provide to all of you. So Austin, why don't you start? Thank you so much.
Sure. Let me bring my slides up. All right, so hello everyone. Thank you all for being here today. So my name is Austin Gamble Jarvi. I am Applications Manager for HiQ Technologies. And as Tony introduced, we are a company that makes EPR instrumentation and provides EPR measurements as a service. And so today what I'm going to be talking about is a general introduction to EPR as it relates to structural biology. So I'm going to talk about a little bit of background and a very light dose of theory on EPR so you can understand what it is and how it works and what kind of information we get out of it. And then I'm going to go through some selected examples from literature to show how EPR can be used in conjunction with common structural biology techniques to give you a fuller, richer understanding of protein and biomolecule dynamics and the full picture of what it's doing. So let's go ahead and get started. So the title of this talk is the conformation gap. And so I want to talk a little bit about what I mean by that to set the stage for what I'm going to talk about from here. So what we mean by the conformation gap is that it's generally known now in the scientific community, the structural biology community, that proteins and biomolecules are they're dynamic ensembles, they're not rigid fixed structures. And that might seem fairly intuitive at this point, but it wasn't always intuitive. And so really the conformational movement, we have these systems that are constantly moving, shifting between conformations, interacting with other proteins, with nucleic acids, with small molecules, with things like that. So really, it kind of puts a nuance on the classic structure function paradigm where that structure and dynamics really define the function. And so, this helps us get a full picture of what a particular system is doing. And so poorly, or not necessarily even poorly, but just not fully characterized dynamics and not fully characterized conformational ensembles can derail drug programs and drug development farther down the line than it necessarily needs to. If you have things like, off-target interactions or some conformational states that you weren't aware of that cause problems later on down the line. So what we're proposing is that EPR is a tool to help prevent that, to either help these programs fail faster so there's not the time and money spent on things that aren't going to work, or to help give you a better understanding so you don't reach those failure points at all. And so EPR is going to help close this gap by directly probing the conformational ensemble of these really dynamic and flexible systems that you can use in conjunction with your other structural methods to more fully understand the systems that you're targeting. And so this presentation is going to show how EPR is used to deliver these insights and how our specific brand of EPR at HiQ Technologies does that. So to start, I'm going to give you a few slides on the background of EPR, on generally how it works in a very high level. So EPR is electron paramagnetic resonance spectroscopy. If you've heard electron spin resonance, ESR, it's the same thing. And so what EPR does is it detects unpaired electron spins. And so you can think of it as conceptually analogous to NMR, which many more people are familiar with. But where in NMR you're detecting nuclear spins, in EPR we're detecting unpaired electron spins. And so that gives a few distinct implications and differences from NMR. But generally, if you think about, an electron, we can think about a spin state as being roughly up or down, and Quantum mechanically, it's more complicated, but just for the purposes of this, when you put those spins in an external magnetic field, you get a splitting of their energy states. So spin up and spin down have different energies, and you can drive transitions between those states by applying microwave radiation matched to that energy difference. And that's effectively how NMR works. That's effectively how EPR works too, just on different particles. And so EPR operates typically at a much higher frequency regime than NMR does. So EPR, the most common frequency bands are about 9.5 gigahertz and 32 gigahertz, whereas NMR typically operates in the megahertz range. And so that gives EPR an intrinsically high sensitivity because you have more polarization between the two spin states. Now, the other thing to note is that in EPR, because we're detecting unpaired electron spins, few biomolecules, proteins, DNA, things like that, have stable radicals incorporated in them. Outside of certain metalloproteins that bind to paramagnetic metal ions, there's not a lot of stable radicals in these biomolecules that we're interested in studying. And so that's a bit of a double-edged sword. On the one hand, it means that EPR is a very specific, selective, and unambiguous technique. The signal that you're measuring is very likely the signal that you want to measure because there's really no background signal in the vast majority of these samples. The other side of that is because there's not native radicals in these systems, we have to introduce them in order to perform EPR measurements. Now, thankfully, that is a very well-studied field, that there has been a lot of great development in the EPR community over the past decades, and you can do this through a process called site-directed spin labeling, or SDSL. And so this is a process by which you take your biomolecule, you can do specific point mutations, and then you can attach stable radicals to those mutations. Most commonly, this is done by cysteine mutation, so you can put a cysteine in, and then the most common spin label just has an active thiol group that can react with the cysteine to form a disulfide bond. It's very robust. It's very easy. Now, in some cases, cysteine mutation can be problematic. If you have a lot of cysteines in your protein, you could get nonspecific labeling if you don't generate a cis-null mutant first, but that can also be a problem if those cysteines are involved in the structure or if they're involved in some function that you can't mutate away. So there's been a lot of development in the field to create alternative spin labeling methods that either have different radical groups that you can detect and different linkers to the protein itself, so that if you have a particular system that has some certain limitations, there's very likely a spin labeling strategy that can get over those limitations and allow you to perform EPR measurements on your system of interest. Now, most of these examples that I'm going to talk about and the context I'm talking about now, I'm saying proteins and relating to proteins, but it's also important to note that this is very easily applied to nucleic acids as well. There's a lot of work done on labeling of DNA and RNA, so if you're particularly interested in those systems, there are spin labeling strategies for you too. So to talk about the type of EPR that HiQ does, I think it's helpful to conceptually break down EPR experiments into a few categories. So EPR, like NMR, is a very broad field. There's a lot of different experiments you could do. There's a lot of different types of information you can get. And so rather than break it down by experiment, I think it's helpful to break it down by the information. And so that can typically be done in terms of how many labels you're putting on your system. So you can either put one spin label on your protein or DNA, and you can get some information on local dynamics, on secondary structure, on the solvent accessibility of your label, on your side chain mobility and molecular tumbling, which is all very interesting information. That's not the subset of EPR experiments that HiQ focuses on. It's something that is accessible to us, but we really focus on when you have a molecule and you have two spin labels on there, because then you can measure the structure, the conformation, and the dynamics of that system. And that's done through a subset of EPR experiments called pulse dipolar spectroscopy, or PDS. So PDS is a subset of EPR experiments that measures nanoscale distances between these two spin labels on a biomolecule. And so through those distance measurements, we can characterize the structure, the dynamics, the flexibility. And so in short, what this does is it isolates the dipolar coupling between these two, your two spin labels, and measures the strength of that dipolar coupling. And we can relate that back to the distance between them. Right, and so on the right-hand side, you can see the 4-pulse deer experiment. I'm not going to go into the depth of the theory of how DEAR works, but this is just there as this is the most commonly used, the most popular of the PDS experiments. And if you've heard about EPR in a structural biology context, you've likely heard of DEAR spectroscopy, if anything. And so you can see the characteristic DEAR signal down on the bottom left, and then it gives you your distance distribution on the bottom right. And that's really the key of these experiments, is that it doesn't just give you a static distance or a mean distance, it gives you a distribution of distances. So this is a probability distribution of all of the distances that you have captured in your total ensemble. So these experiments are done at cryogenic temperatures. So you take your labeled sample, flash freeze it, and it captures all of your conformational ensemble at once, and then you measure it, and EPR reports on all of the conformations present in that ensemble. And so the shape of the distance distribution is really important and really powerful, because that's how you can determine what's going on in those conformations ensembles. So if you have a very narrow distance distribution, that implies that you have a very rigid conformation, that the spatial distribution that your spin labels can occupy is very narrow. But if you have a very broad distance distribution, that implies a lot of flexibility, that your subunits can move around a lot, and so your spin labels can occupy more space, so your probability distribution is broad. And in the case, the interesting case where you have multiple conformations present, multiple distinct conformations present, you can see that manifesting as multiple peaks in your distance distribution, and you can get this type of information out for all of these different conformations. You could track population levels, you can watch how they react in response to different stimuli. It's a very powerful technique, and so through the next few slides, I'm going to show you how you can use that in combination with other techniques to really fill out your understanding of your protein system. And so at HiQ, we do this through FATHOM, which is our automated EPR system. So this is all in one, everything you need to run these EPR distance measurements. And so we've designed this with an emphasis on sensitivity and stability, so that you can measure low concentration samples, low population states, and that the instrument itself is stable enough to run for the periods of time necessary to collect all that data. We've put a high emphasis on the user experience, so that you don't need a background in EPR, a PhD in EPR to run this information. It's designed so that technician level users can go up to it, enter some information, and get their distance distribution at the end in a nice, well thought out report. We've designed it for enhanced throughput by adding the ability to put in multiple samples, 5 at a time in one go, instead of a single sample in a single tube in, and that tube out, rinse and repeat. So now you can put 5 in at one time and let them run back to back to back to back, all automated. And we've tried to make it practical and emphasize the utility and allow it to characterize a broad range of biological samples in this way. And so there's a wide range of applications for EPR, even just in the structural biology realm. And so this is what I'm going to talk about over the next few slides. I'm going to get into some specifics, but just generally, we can see things like EPR works really well with Cryo-EM and AlphaFold. It can help refine models and confirm predictions. It's useful in characterizing things that are notoriously hard to characterize, like IDPs, like GPCRs, like in-cell samples. And it, as I mentioned, it can be applied to things like DNA and RNA. It can probe nucleic acid protein interactions. So there's a wide range of things that EPR can be applied to. And so with that, let's get into some specifics. I've got 5 or 6 specific examples that are pulled from the literature to emphasize how EPR works well with structural biology workflows. So this first one is a nice prototypical example of how EPR can be used in routine structural biology workflows with cryo-EM structures. So cryo-EM is obviously a great technique. It's really become one of the industry standards in recent years. And so it's great at providing, high resolution structures, but there are some points where it can struggle with, namely in flexible or disordered regions where the resolution is low. And so a nice example of that, where this group had a nice cryo-EM map of a particular protein, but they were interested in some conformational changes that were occurring, they suspected were occurring in the circled red region. And unfortunately, that region had a low resolution from their cryo-EM data, low enough that they couldn't really resolve what they wanted to resolve. And so in order to get a full picture of what was happening, get the information they wanted, they turned to EPR, performed some strategic labeling within that low resolution area, and did these types of distance measurements. And you can see that information on the right-hand side of the slide, where you've got two columns. The first column is the experimental data we'd pull out of the EPR measurement, and then the distance distribution that's extracted from that data. And you can see this is all the same protein, the same labeled sites, just they've added different ligands to the protein. And you can see, there are shifts in the distance distributions and differences in the distributions in response to different ligands being binding. And there's even differences. They've got some dashed gray lines that indicate what their predictions were from existing structural models. And so what this is telling you is that EPR can be used to, in conjunction with these structural models, to refine your understanding of the conformational shifts in response to various ligand binding, and to fill out your understanding of what conformational states actually exist when these ligand binding, when these functions actually occur. So this is a really nice example of how EPR can help supplement and complement great structural data. In a similar vein, it works very analogously with something like AlphaFold, where, again, AlphaFold has been revolutionary in terms of protein structural predictions in recent years, but it also can struggle with flexible regions with intrinsic disorder, and it'll spit out regions of, low confidence. And EPR, much like it was used in the previous example, can be used to refine those regions of low confidence by providing point-to-point distance constraints that you can then feed back into your AlphaFold algorithm to help refine the model. You can also use EPR distance measurements in something developed called DeerFold to help spit out or generate a wider range of different conformations. AlphaFold is generally good at giving you a structural prediction, but is not great at giving you conformational ensembles. But Deer constraints can be used in conjunction with AlphaFold algorithms in order to help generate those missing conformations. So there's a lot of nice synergy with AI prediction as well. So from here, instead of talking about synergy with techniques, it's more going to be about how EPR is applied to specific classes of biomolecules. So first up, we have GPCRs, G protein coupled receptors, which are a notoriously dynamic and often difficult sample to measure structurally and dynamically. So GPCRs are involved in a whole lot of physiological and biological functions. They're a very important target for drug development. And a lot of that importance is in their conformational modulation in response to various, ligand bindings or protein interactions, things like that. And so we have two examples. On the left is the GPCR angiotensin that is this group took and labeled at a number of different places and measured the EPR distance constraints in response to binding different ligands, different agonists. And you can see that there's differences between the apostate and the different binding states, and in a lot of these different transmembrane helices and a lot of these different subunits. And so using this information, they can combine that with existing structural models to get an idea of how these conformational changes actually occur, what things are actually happening, the general flexibility of these different conformational states, and track that through the mechanism of ligand binding and the function of the GPCR. So it's a really powerful tool for defining, mechanisms and complicated conformational changes. On the right-hand side, this isn't necessarily exclusive to GPCRs, but this is a nice example of how EPR can be used to track oligomerization states. So in this particular example, this was a type of rhodopsin that this group was monitoring how it oligomerized in response to pH change. So you can see at low pH, there's no oligomerization, and then at higher pH, there is oligomerization of these GPCRs. And they did that by labeling, putting one label per protein, and then as the pH increases and these proteins start assembling, you can see the distances start to appear. So when the pH is low and there's no oligomerization, there's no distance because you only have one spin label. But as they start to assemble, you get multiple spin labels, and so those distances start to appear. So this is a really nice way that you can track oligomerization states. And again, this isn't exclusive to GPCRs. You can do this to really anything that forms at least a dimer. You can use this to track that sort of assembly. So another notoriously difficult to characterize system are IDPs, intrinsically disordered proteins. They're difficult to characterize structurally because they don't just exist in one ordered structure, they exist in many structures. And so that's what this group was looking at. So they took tau fragments of the tau protein, which is involved in things like Alzheimer's, and they performed cryo-EM on these tau fragments and got a structure. And then they also performed EPR distance measurements on these structures. And so from the cryo-EM structure, they generated what the anticipated distance distribution would be for the EPR measurements. And you can see that in the bottom left. But then when you compare that to the actual EPR measurements, you see that there's a whole range of probability distribution that doesn't exist in the cryo-EM information. And that's showing that EPR is capturing the full set of conformational states that cryo-EM isn't sensitive to. They did the same thing with AlphaFold, where they had AlphaFold generate a few different possible structures for these tau fragments. Did some simulations and modeling to determine what the proposed EPR distance distributions would look like, and then compared that to the actual EPR distance distributions. And it's the same story where there's a whole set of conformations that aren't captured by AlphaFold, that aren't captured by Cryo-EM, that EPR has given you insight into. So it's just another great example of how generally, EPR can complement these existing structural methods to give you the full conformational ensemble, and it's also showing you that EPR is a really powerful tool for looking at IDPs that are very tough to pin down by other methods. In a little bit of a shift, this is a nice example of EPR tracking disease states in neurodegenerative diseases. So this particular group was looking at SOD1, which is a homodimer that has one endogenous copper-2 binding site per monomer. And so that means when this protein dimerizes, you have one copper per monomer, you have two coppers, and you can measure the distance between them. And so SOD1 is involved in ALS, and there's certain mutations specifically of SOD1 that are involved in ALS. And so this group showed that by doing distance measurements on wild-type SOD1 and then SOD1 mutations, they could actually distinguish between mutation states that are linked to the disease state of ALS just by looking at the distance distribution. So you can see in the data on the right, when this protein is associated with a mutation that's associated with ALS, the distance distribution broadens. So EPR can be used as a nice diagnostic tool to detect certain disease states as well. And so as the last, I talked a little bit about labeling nucleic acids. As the last example, nucleic acids and CRISPR complexes are also a huge topic, and spin labeling strategies can be easily used to incorporate spin labels into these nucleic acids, and even on proteins and nucleic acids at the same time to probe the interactions between them. This group specifically took a DNA associated with a CRISPR complex and labeled each individual strand of that DNA. So when they duplex, there's two labels, and they could use EPR distance measurements to track its life cycle through the various states. So you can see on the bottom left, from free DNA, just unbound, floating around in solution, to DNA that then is bound to Cas9, and then as that DNA gets cleaved, you can see more shifts in the distance distribution, indicating that, we've got the separation of the two strands of the duplex. And so this is a great way that shows how EPR can be used to track, nucleic acid mechanisms as they go through the CRISPR process. And just as a nice supplement, this group also performed MD simulations and compared those to the EPR distance measurements. So we have some broad constraints from EPR and insight into overall flexibility and dynamics. And then when coupling that with something like MD, you can get a really fine atomistic insight of what is actually going on. So it's a really powerful combination as well. So hopefully over the last few slides, I have shown you that EPR is a really powerful biophysical technique. So the core here is that we really want the best understanding of our systems as possible, right? We want to know as much information about them as we can to make informed decisions about, targeting, about certain drug development programs, etc. And so the best information comes from using a lot of complementary methods. So we've got Cryo-EM, X-ray, NMR, all commonly used in structural biology today. We've got computational tools like MD. We've got AI and AlphaFold predictions. And now we'd like to introduce EPR into that workflow. So EPR can help supplement these structures with that missing dynamic dimension through these long-range distance distribution measurements. And so we think as part of an integrated workflow, EPR can help confirm conformational states and conformational ensembles, and can help refine and validate models. And overall, it can help ensure you have the most information possible for drug programs, and so that they are built on biologically relevant structures and conformations. And so with that, that's all I've got. So I'm going to pass this back over to Tony to talk about, their offerings at Creative Biostructure and how EPR fits into their broader portfolio. So thank you all very much.
Thank you so much, Austin, for the detailed technique presentation. I'll take it from our side. Again, I'm Tony Zhang, and Senior Project Manager and Principal Scientist at Creative Biostructure. We're thrilled about this collaboration and being able to offer quantum-enabled EPR as part of a service platform. And this is something our team has been looking forward to. Austin has covered the technical details with real data, and I like to provide an outlook of how does a technique like this fit in our CRO portfolio at a moment where there's a great deal of enthusiasm about AI in biology. In my opinion, we are in the pre-AI era of structural biology. Lots of AI pipelines are sprouting, claiming that they can solve real-world problem. And in the end, we always find there's a problem of short of qualified data in the field to train good AI models. So we're in the need of considerably more clean, well-characterized experimental data than we currently have. And it's part of why high-Q FATHOM EPR interests us. A brief word about us first. Creative Biostructure is a structural biology and drug discovery CRO. We work across both the wet lab and dry lab. On experimental side, Cryo-EM, NMR, X-ray crystallography, protein production, including difficult targets like membrane proteins, protein complexes, and their functional and biophysical characterization. On the computational side, a structural prediction with in-house built AlphaFold and a CoLabFold, molecular dynamics, docking, structural-based design, data processing. We also facilitate in publication and deposit of PDBs. We are running a summer promotion currently with a focus on structural biology. So let us know if you need any help with your projects, and we can offer generous discounts. The computational side has become much faster and much cheaper recently. Structure prediction that used to be a substantial undertaking is now routine. But it's worth being precise about why that worked. AlphaFold is a genuine achievement. It was also made possible by Protein Data Bank, 50 years of carefully curated, standardized experimental structures, but by machine learning standard, the PDB is a small data set of about 250K structures, and nearly all of them are static. So when prediction struggles with conformational ensembles, with disorder, with anything that moves, part of the reason is simply that there is no equivalent of PDB for dynamics. That training data doesn't exist yet, and we wish there were. And that's what I mean when I say we're in a pre-AI era. Note that the models are not that the models are poor, they're remarkable, but the data they needed hasn't been generated. And I'd also note that the major AI companies appear to have reached a similar conclusion. They invest now, in high quality experimental data, not only into more computing powers. So it's worth asking where our experiment method leave the gaps. Crystallography gives us superb resolution, but it needs protein to hold still. Flexibility works against it. NMR report on dynamics and does it well, but it's limited by molecular weight. Past a certain size, it becomes very difficult. Cryo-EM is where most of the attention is, and deservedly so. But anyone who has processed a data set knows what happens to flexible regions. They blur, they average it out. Sometimes they simply aren't resolved. So there's a real gap, and the EPR feels it. EPR doesn't replace anything on that list, but it produces a kind of information the other methods struggle to provide in the form a model can actually use, and there is practical reason for that purpose. EPR has historically been slow and sample intensive, which is exactly what FATHOM is built to solve. So let me be concrete about what FATHOM EPR brings to our service portfolio. Cryo-EM, NMR, and crystallography tell you what a molecule looks like. EPR tells you how it moves. On the left, static structure is one average snapshot, and function often depends on exactly the motion that gets averaged away in many cases. On the right, the deer gives you a distance distribution between a pair of spin labels, roughly 2 to 8 nanometers, a distribution then rather than a single value. So you're seeing the ensemble, not the mean. No molecular weight ceiling and no crystal required. Now, each measurement reports on one pair of sites. That used to be practical sitting. Every additional pair meant another slow experiment, and you accumulated distances one at a time. With the throughput Austin described, that arithmetic changes. You can label several pairs across a protein and measure them in a much more reasonable amount of time. And a set of distances across a protein is a map where the domain sits relatively to one another, which states are populated and in what proportion, how the ensembles shifts when a ligand binds. The labeling itself takes some thoughts, and I'll come back to that because it's where this collaboration matters most. So EPR complements other methods. It reports precisely what other methods find hardest, and it does it in a form of a model can use. And on the AI side, Austin has mentioned it, and here's the framing I find useful. AI predicts, the simulation hypothesizes, and EPR measures. Consider how this program typically runs now for AlphaFold. You start with a sequence, and it gives you a structure quickly, cheap, most of the time accurate on compact targets. You run molecular dynamics, the simulation propose how the structure moves, perhaps. You can then dock a ligand or design a binder. But if you step back, nothing has been measured yet. For a well-folded rigid protein, that's often acceptable. The prediction is confident, and the force field is well parameterized, and the results hold up. But many of the targets that matter most aren't like that. GPCRs moving between conformational states, intrinsically disordered proteins, ternary complexes with considerable freedom, and cryptic pockets can open transiently. Those are the regions where the model is least confident, and in practice, they are rarely checked experimentally. That's the gap we are interested in. An EPR can put a measure number on the region where previously you had only a prediction. So, and Austin walk you through the methodology in this particular paper. So let me instead say why I think it worked as well as it does. There is a useful coincidence here. The regions where AlphaFold is least certain are largely the same regions where EPR is most informative. And that gives you three things. You can check the low confidence regions. The low PLDDT score is the model reporting accurately that it doesn't know. AI measures a measured distance across that region tell you which prediction was correct. You can distinguish between states. Prediction increasingly returns several plausible confirmations without a way to choose. EPR indicates not only which states are present, but approximately how populated they are. And you can use the distance as constraints that, the Dierfold approach. What I found noticeable is that the workflow, how little data was required. A small number of well-chosen distances was enough to guide the model toward a correct confirmation. So that's the point I want to emphasize. A small number of distances goes a long way, and each one constraint that contains the relative position of the two entire regions of the protein. So placed where the model is uncertain, and a few measurement collapse a large part of the confirmational search space. And then, the high throughput, changes the fact that you no longer are limited to a few options. But the practical difficulty is that running this loop requires prediction simulation experiments. Those were usually three different providers on three different timelines. And this is exactly where what this collaboration is intended to address. I'd like to spend a few minutes on the practical side because I think is where our perspective is most useful. The first is where the labels go. So you don't label wherever it's convenient. The sites have to report on the motion you actually care about, which means the scientific questions needs to be settled before construct is ever made. Functionally important residues are off limits. Buried positions are a problem because if a site isn't solvent accessible, the labeling will have a low efficiency. And the native systems have to be accounted for. Sometimes they lead to the aggregation or denaturation of certain proteins, and the thiol chemistry cannot distinguish the ones you intended. So choosing those sites well require EPR experience behind it. Site selection tends to become a trial and error. If you build a construct, you measure, you learn something, you build another one, and that is slow and it's expensive. This is the clearest place where a collaboration like this earns its keep. Austin and his colleagues advise on the label placement, drawing on actual EPR experience. We do the protein engineering. And the second is where the protein tolerates any of it. And here I can speak with more confidence because this is our routine work. The construct has to survive system engineering, and sometimes it doesn't. Removing native systems can sometimes destabilize a protein, like I said. It has to remain stable at the concentration that the measurement requires. It has to remain folded and functional after labeling, which is not guaranteed. Membrane proteins make every one of these harder, and difficult samples are large part of sample we do. And I'd say that this is the most where the most structural biology projects are won or lost, which leads to my next point. So producing typical proteins, properly folded, well-behaved, and modified wherever you need them is a reasonable description of what we do, rather other than structural and physical works. So first of all, memory protein expression, toxic and difficult targets, including GPCRs, expression system chosen for them rather than adapted to them. And then we can produce memory proteins in different forms, especially nanodisk reconstitution, which provides native-like lipid environment. So you measure what you measure, it reflects the protein in a membrane rather than in detergent. And then protein labeling and construct engineering, site-directed mutagenesis, cysteine engineering, and conjugation chemistry are routine work for us as well. Spin labeling is a specific application of methods that we previously run. And for targets where cysteine chemistry isn't a good fit, there are other routes like cell-free expression. They allow non-canonical amino acids to be incorporated during the translation, which places a label at the position originally file chemistry might not reach, including insertions, so including sites that becomes buried once the protein folds. So for difficult memory proteins, co-translation, insertion into nanodisk is possible. But that demands work. It's exactly the kind of problem this collaboration can be built to take on. And that's the shape of the collaboration, and I think it's a very clean one. HiQ brings the instrument and the EPR expertise and the judgment about where the label belongs. And then we bring the protein correctly engineered and then correctly labeled. With that, I want to put it together, the pipeline or the workflow. So first, express and label of the protein. So HiQ will provide us on label placement, and then we engineer and produce the protein. And then on the FATHOM EPR measurement, the automated adhere will require only microliter of volume, and then it has very fast measurement compared to conventional EPR. And then we, as a company, we integrate those distances measurement with the Cryo-EM density or AlphaFold models, and then MD trajectories, as we already run them in-house. And then we will deliver our customer a report of the structure and dynamics and what this means for your protein or other macromolecules. So if your target is flexible, disordered, membrane associated, or simply hard to pin down, those are the cases we want to hear about. And thank you so much for your attention, and we are happy to take any questions. Thank you.
So with that, I would like to thank everyone for your attention. And now we have a Q&A session for everybody to ask questions. Thank you so much. Okay. All right, so this customer's asking if I'm a customer approaching you for a project, how will the process look like and how long will the process take? So we will first have a meeting with you and talk about your actual need and if you have your protein of interest, and then if you have a partial structure or AlphaFold structure, we will help we will help you generate several mutations. We're taking care of the, regions where, the most of the flexibility happens. And then we will design, pairs together with FATHOM EPR using their expertise and give you, in the form of, plasmids and then express them and then label them and measure them. So those process we usually take a little bit more care, not a little bit more, but more care in terms of planning, because as I said in the presentation, is where the projects fail or win. And then nowadays we have many computational and more advanced wet lab strategies to solve these problems. And together with the speed of the FATHOM EPR, we're confident in solving many of the difficult problems of protein or macromolecule flexibility. How long will the process take? It usually takes, 2 to 3 weeks after we design the protein pairs. That could mean that if you have 1 to 3 plasmids of different mutations, we can finish from expression and purification within 3 weeks. So and the whole project, depending on the difficulties, I would say from weeks to months. But then this process is highly accelerated nowadays because we require less amount sample and we have faster measurement. Thank you. Any other questions? Okay. One. So okay. So if no more questions, I would like to thank everybody again and for your attention. And if you have any other any projects involving protein or macro or DNA or macromolecule flexibility, please find us, and then we are ready to help. Thank you so much, and have a nice day. Thank you.